Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell, going to be your host here for this episode of the Real Science Exchange. Tonight's episode is going to be just a little bit different. At the 2024 ADSA conference, Balchem presented 8 abstracts during the scientific sessions. And tonight we're going to record short segments with each of the presenters or the researchers, student researchers for each one of those abstracts, along with their advisors. And then we're going to put them together in one podcast. Now, this allows us to do two things, right? It allows us to kind of showcase some of the rising stars in the industry. And here at Balchem, we just love to do that. And it also gives us an opportunity to showcase our research. So enjoy.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
And in this segment, Clay and I will be reviewing a poster that was given at this year's ADSA, and the title of that poster was Evaluating the TMR Stability of Rumen-protected lysine Products. That poster was presented by Kari Estes, and her advisor was Dr. Mark Hanigan. Now, Dr. Hanigan, I don't believe this is the first time you've been in an advisory role for Miss Kari. Is that right?
Well, I'm not even sure what year it was, but she started working in my laboratory as an undergraduate student, I believe a sophomore. And she worked on some of the techniques we were trying to develop for assessing bioavailability of amino acids at that time. And then she decided that the project she was helping the graduate student on could be done better. And so as an undergrad, she then worked on it some more. And then she worked as a technician for a year, I think. And then we got Balchem to fund her, as a graduate student for, a couple more years. And then we sort of hung on to her a little bit, because she'd come and help us for a few years. And
Kari, why don't you just give us an overview of the poster that you presented there at the ADSA?
Yeah, so with any rumen-protected nutrient, there seems to be a lot of focus on rumen protection and bioavailability or intestinal release, right? But I think a piece that is or can be crucially missed is the feed or the TMR stability of these products, right? That's going to be the first environmental challenge that the products can incur. The moisture and the acidity perhaps of the TMR could be quite damaging to some of these products. So we wanted to evaluate 5 commercial rumen-protected lysine products and a Balchem prototype for TMR stability. And we didn't develop the technique by any means. It has been published already in the Journal of Dairy Science from the Minor Institute. But we did make a modification to it, which was adding a positive control treatment, which was an unprotected lysine treatment.
I want to point out as well, so we don't lose sight of this, this poster was actually picked for Spotlight. The committee picked, a few of these favorite topics, and it was also presented as a Spotlight where Kari did a 3-minute summary of the poster in front of an audience.
Yeah, so I tried not to do an oral presentation, but I still got one out of it.
Exactly.
Yeah. So, so if you want, I can briefly tell you how we ran the experiment.
That'd be awesome.
So we took a sample of TMR, and we took the equivalent of 1 gram of lysine from each of these products, right? The products ranged in protection technology and lysine content. So we took that into account by equalizing that piece of it with the 1 gram of lysine. So we gently mixed them together for 30 seconds, put them in a Ziploc bag, and they sat there for 0, 6, 12, or 24 hours. And then at the end of each time point, we took the contents, put them in a strainer bag, just dipped them in water, distilled water, for 1 minute, allowed them to drip dry for 2 minutes, and then we took a sample of the solution and had that analyzed for free lysine content. We had that done at a third-party lab. Yeah, so it's a very quick procedure, which is nice, but yields some very interesting results for sure.
Very quick, but very labor intensive for that period of time, correct?
That is true. That is true. Yes. When you're talking 1 or 2 samples, it's very quick, but that number jumps up pretty quickly when you run everything in triplicate. So yes.
Kari, why did you pick the time points that you selected?
We picked those based on the typical, maybe not typical, but potential feeding times on farm. The 24-hour time point would be a 1x feeding. The 12-hour time point would be 2x feeding. And then the 6-hour time point would be the 4 times feeding.
Thank you. So Kari, you want to discuss the results?
Yes. So interestingly, with the unprotected lysine, right, that was our first objective, right? Are we able to recover all of that free lysine that we put into the TMR? And we found, much to my surprise, that we were unable to recover all of it. And as the incubation time increased in the TMR, the recovery actually decreased. So even at time 0, right, so that was just sitting in the TMR for 30 seconds, we recovered about 85% of the free lysine. And then by 24 hours, it was down close to 50% recovery.
So Mark, do you have any thoughts around what might be occurring where we're not recovering all of that, all of the unprotected lysine HCl?
Yeah, I mean, feed has a lot of different compounds in it or components, and a lot of those molecules that are in there have charges on them, and so they're able to bind to something like lysine, which is positively charged, right? And so if there's a negative charge anywhere on that feed, it's going to bind lysine and not want to release it again very readily when you go to wash it. And we know there's lots of binding sites all over feed. That's not a new finding. So it's just soaking up a bunch of the lysine that gets put on it, and it's not releasing it very readily.
Thanks. So Kari, what were your findings with the rumen-protected lysine products?
Yeah, so after we found those findings with the unprotected lysine, we did create a correction factor that we applied to the data for the rumen-protected products, right? Because any of the lysine that they could be releasing could undergo that same fate as the unprotected lysine. So once we applied that correction factor, we saw that there were very different stability values among these commercial products. So ranging from, there was one product in particular that released close to 87% by the 24-hour time point, versus the other extreme, we had another product that had much lower release, which was only 9% at the 24-hour, and all of the other products kind of fell in between that. So quite large differences between the products.
Kari, I'm curious if you have a theory as to the ones that were not well protected. What might have caused that? Was it something in the structural design of the end cap, or any thoughts related to that?
Yeah, that would be my first inclination, would be that the encapsulation or the protection technology maybe is not as good as other ones. So that moisture or acidity coming from the TMR is just able to break down that coating and reach the lysine more easily than others.
All right. Very
So
well.
is that related, do you think, the solubility too of them or not? I mean, because those products have different A fractions, so to speak, right? The soluble fraction that's readily available in the rumen.
Could be, yeah, especially at that 0-hour time point, for sure. Mm-hmm.
So, Kari, how do these findings compare to, you mentioned at the beginning there was some previous work done and published. How do these findings relate to that previous work?
Yeah, so I think there were maybe 2 products in our experiment that were also tested in that published paper,
Yep.
and they matched very, very similarly. Yep.
So what about, is it possible that some of these products could have passed through the ag when you were doing the protocol?
That's a very good question. So the strainer bags that we use are a 250 micron. So I will say visually, I didn't see anything come through that bag, right? But the very last step of taking the sample of the solution, it is filtered in a 0.45 micron filter. And I can guarantee you that none of the products were going through that filter. Yes, very, very fine. Yeah.
What are the implications of this potentially for nutritionists and dairy producers?
Yeah, so I think based on this data that what we found is that we definitely need to be taking into account TMR stability when we are trying to determine feeding rates, or even how we should handle some of these products, right? Maybe some of these products we need to handle differently, one versus the other. But they'll all be in a TMR, I guess, eventually. So, yeah, we just need to take it into account for sure.
And maybe something people need to think about, right? As they're presented with research, did these products actually go through it? Were they fed in a TMR?
Mark and Kari, thank you very much for joining us today. In this episode, Clay and I will be reviewing a companion poster to the one that you just previously listened to. And the title of this one is Evaluating the TMR Stability of Rumen-protected Choline Products. Again, that was done by Kari Estes, and Mark Hanigan was in an advisory role. Kari, wanted to start us off with kind of giving us an overview again of why is it important to measure the stability in a TMR, right? When we think about rumen-protected products, we're thinking about protecting them in the rumen. Why is it important to protect them in the TMR as well?
Yeah, I think it's a piece that can be missed for sure. These products can sit in a TMR for up to an extended period of time, right? Perhaps 24 hours. And so we need to be looking at the stability of those products before they even make it to the rumen, right? They could be damaged completely, even for all we know. So,
right. Yeah, we always say, right, the three-legged stool of a good ruminant end cap: feed stability, ruminal stability, and intestinal digestibility. You truly do lose the game from the start before the cow ever eats it if you're losing it either in feed mixing or in the TMR.
Yeah, good point. Kari, why don't you kind of give us an overview of the objective of this study and what was the protocol of the study?
Yep, so we wanted to look at the TMR stability of 5 commercially available rumen-protected choline products. And this one was a little bit different, right? We did lysine before, or in our previous poster, but as I had mentioned before, that technique had been completed before and published in the Journal of Dairy Science by the Minor Institute. But choline had never been done before. So we really just wanted to see if this technique would work for choline. We figured it probably would because it seems to behave pretty similar to lysine in terms of the hygroscopicity of the nutrient, but we figured we would give it a whirl. So we still did make that modification, though, where we added the unprotected choline chloride to make sure that we were able to, well, not even to make sure, we just wanted to see how much we'd be able to recover, right? See if we were able to recover 100% of it at the different time points. So the protocol was still the same, where we took the TMR and the equivalent of 1 gram of choline from these products, mixed it together for 30 seconds, let it sit in a Ziploc bag for 0, 6, 12, or 24 hours. We put it in a strainer bag, dunked it in 1 liter of distilled water for a minute, let it drip dry for 2 minutes, and then we took a sample of the solution, filtered it, and analyzed it for choline content. So very similar protocol.
So, Kari, maybe explain again why those particular time points?
Yes. So for the, we picked the 24-hour time point because that would mimic a 1x feeding per day. We picked the 12-hour for the 2x feeding per day, and then the 6-hour for a 4x feeding per day.
And certainly, with pre-fresh diets, these close-up diets, it's very common to only feed them once a day. So that 24-hour time point is pretty critical, especially with rumen-protected choline. So, Kari, what were your findings here?
Yeah, so again, for our first objective, that was looking at the recovery of the unprotected choline, and we saw very similar results to the lysine in that we weren't able to recover 100% of that choline at any time point. And it mimicked very similar to the lysine in that we recovered close to 80% at the 0-hour time point. And then as incubation time increased, the recovery decreased. And then we recovered close to 50% by the 24-hour time point.
So Mark, would you care to speculate on why we did not get complete recovery of the unprotected choline chloride in the TMR?
Yeah, as we talked about before, it's binding sites that are all over that feed, and particularly when you cut it up, and open up the inside of the cells and stuff. I mean, there's all sorts of things that are going to bind to choline. And so it would have been really surprising if you got 100% recovery. And I think looking at some of the stuff that's in the literature, I mean, they failed to make that, assessment, right? It's not that this is bad that it happens. It's just something that we have to correct for in the assay. And if you don't correct for it, you're going to way overestimate the loss during your TMR stability test.
I was wondering as well, and maybe the 24-hour one, would it be possible that we might be seeing some microbial activity or fungal activity that might be degrading the nutrients or at that time point? No thoughts?
I'm not sure you're asking, but yeah, I think that's a possibility as well, particularly if you're leaving it for 12 and 24 hours. That's a long time for a microbe. So yeah, there's lots of microbes in the feed, and they could be chewing it up as well if it's free. If it's protected, then presumably not. But obviously, our reference point is the free concentration.
So, Kari, what were the rest of the results that you found on the rumen-protected products?
Yeah, so again, like the lysine, we saw a very vast difference in the stability of these products. So at the 24-hour time point, we had quite the spread. There was a product that had 100% release by that time point. And then we had a product that was about 5% release at 24-hour. And then all of the products, the other products were in the middle of those two. So quite the large spread there.
So what would you attribute those differences to?
I would say the protection technology or the encapsulation technology that is utilized on these products, for sure. Some of them definitely are providing more protection than others for that moisture and potential acidity or microbes to get through that coating and actually get to the choline.
And choline chloride itself is it's very soluble, correct?
Yes.
So is some of that difference just due to solubility in the encapsulated products then?
Yeah, I would think so.
So, Kari, what are the implications of this?
Definitely, as I'd mentioned before, I feel like it's a component of rumen-protected products, not even just of choline, right? That can be overlooked. We focus a lot on rumen stability and intestinal release, but we need to be evaluating these products for TMR stability, because this is going to be the first environmental challenge that these products are going to endure, and that's even before the cow consumes it. So we need to be looking at this piece very critically.
So, Kari, what's next?
Well, I definitely want to continue testing lysine and choline products as they come onto the market, right? New products, but I definitely want to modify this technique to be able to test rumen-protected methionine products. That's my next goal. Maybe next year at ADSA.
What about stability in the mixer?
That's something that we've looked at before. We have a small batch mixer, and we've done some mineral mix stability testing. And that also has some very interesting results based on product and encapsulation technology.
Yeah, and I do want to add that these both the feed mixing protocol and now the TMR stability protocol. That's a lot of Kari does this testing as we're developing new products as well. So that that is all part of our product development process. Is looking at feed and mixing stability.
Yeah, good point. Well, Mark and Kari, I want to thank you very much for coming and discussing this very important topic. And in this segment, we're going to be reviewing an oral presentation that was given at the 2024 ADSA meetings down in Florida. The title of the presentation was Effects of Dietary Rumen-Protected Ruminally Infused or Abomasally Infused Choline Chloride on Milk, Urine, and Fecal Choline and Choline Metabolite Yields in Lactating Cows. That presentation was given by Charlie Yu from Cornell University. Charlie's here with his advisor, Dr. Joe McFadden from Cornell. And give us the treetops. What exactly were you trying to understand with this study? And then maybe just kind of a little bit of how you went about that.
Now, we had a really big question, I think, that stands, and something that's very difficult to study is choline bioavailability, and to understand how much choline is used by the animal for different functions. I mean, it's such a complex metabolite in the sense that, the animal can convert that choline into a variety of other metabolites. And current evidence suggests that stage of lactation and it certainly influence choline utilization. So we really wanted to try to better understand that, to try to understand choline use in the cow, and then try to provide some estimates, or at least work towards a way to better understand bioavailability for choline. Now, this particular study is one of two studies that we performed in recent time here to try to assess this. And this was more of the economical way of doing things. I'm sure that there are a variety of approaches to look at choline bioavailability. But in this particular case, we had 8 rumen cannulated cows. And then we sort of had two groups. They were either blocked as either being late lactation or being early lactation. They differed by roughly 150 plus days in milk in terms of their difference. They were in a 4x4 Latin square, so fairly minimal study design in terms of having just 5-day experimental periods. A lot of questions about is 5 days of a treatment long enough? And there's been consistent studies now to show that by day 4, day 5, you can get a plateau in some of the choline responses. And so, yeah, we think it's more than adequate. And there's been other studies that have shown that a couple-day washout after is more than enough to get things back to baseline. And so we had these 5-day experimental periods, and we had sort of 4 treatments. We had a 0 grams per day of choline chloride. That's our control. And then we provided unprotected choline chloride at 12.5 grams of choline ion per day. And that was infused directly into the rumen as a continuous infusion. And then we had a rumen-protected choline, also providing 12.5 grams per day of choline ion per day. And that was delivered as frequent meals throughout the day. And then we had an abomasal infusion, also providing 12.5 grams per day of choline ion, delivered as a continuous infusate. And they were fed a typical sort of Northeast diet. That diet, though, in terms of their intake, was matched to their baseline. So we didn't want to see the daily sort of variations that you can see in intake with such a small population. So we took their baseline intake, and that's what we basically fed them for the next 5 days and throughout that experimental period. And that's what that does is it really helps stabilize sort of baseline choline and choline metabolite levels in the different pools. It does help to sometimes see differences. And, we did measure milk production and composition, but this was such a small study population. I don't think we can infer too much from that data. But, yeah, we did collect the urine. We collected the feces, and we sampled milk, so we knew their daily milk yields. And the goal there was to try to, again, look at the choline and choline metabolite concentrations in those different pools, in the fecal, the urine, and in the in milk, and also plasma. I think these, the 2 bioavailability studies that we've done, this was being one of them. We're looking at urine more so than we have in the past. I think we like to think that something that's bioavailable, once it gets absorbed, it's used by the animal, and that's it. And I think we can't ignore the urine after these 2 studies anymore. So maybe we can get into the results.
That'd be great. And Charlie, would you like to kind of walk us through some of the main results you got from the study?
So first of all, it's evident that choline bioavailability, it was influenced by the delivery method, like how the choline was fed, either unprotected, either rumen-protected, or either abomasal infused, and also lactating stage as well. And long story short, fecal and milk choline concentration was only observed in early lactating cows only with abomasal infusion. And also abomasal infusion increased some of the choline metabolites like phosphocholine and the fecal and the urine betaine, and but not the end product of choline metabolism like phosphatidylcholine. The end product, it was not changed, and it simply suggests there are potential saturation of choline metabolism in the lactating cow. And also, like in late lactating groups, we saw that supplement with abomasal infused, that it increased the fecal TMA and urinary TMAO levels.
Yeah, I guess we'll add to that and say that a couple of findings that I thought were interesting were that, again, the choline response that you see in milk was evident in the early lactation group. and not in the late lactation group. I think that's really important to emphasize. It's something that was sort of supported by the increase in phosphocholine as well. And so phosphocholine and choline concentrations were enhanced in early lactation and were not so in late lactation in response to the Abomasal choline infusion. I think that sort of speaks to how the high choline demand that the mammary gland has during early lactation. It's really something we've seen repeatedly. We definitely keep seeing the betaine response. I always get this question like every 6 months, like, oh, what's the metabolite that should be the most responsive to choline? It's betaine, okay? No surprises there. And we had substantial increases in betaine. And when you looked at the urine, you could really see that, yeah, that goes into urine eventually. It does go into milk too. But yeah, the urinary betaine concentrations went up really, really high. We were hoping to try to see, in this particular study, we didn't see any response in milk TMAO. So TMAO is a biomarker of gastrointestinal choline degradation. But when we looked at fecal choline metabolites, I don't know if this has been shown before, but maybe we're the first. But the treatment of the choline to the abomasum, so this is post-ruminally, keep in mind, we did see an increase in fecal trimethylamine concentrations. And so, what I want to emphasize here, and our bioavailability studies are showing this, is that there is post-ruminal degradation of choline to trimethylamine. And there are bacteria in there that have an enzyme called TMA lyase, and it will convert choline to trimethylamine. And that would certainly negatively impact the bioavailability of choline. Our other study does, I think, a better job at trying to provide some estimates of that degradation. But here we're looking at sort of single-digit kind of percentages in terms of how much choline is degraded post-ruminally. But we need to be able to understand that, because there's certainly a use of in vitro tools to estimate intestinal digestibility, and those assays don't necessarily account for, or they don't account for, the role of TMA lyase.
So if you're finding TMAO in the cow, do you know, is that coming from ruminal degradation or post-ruminal degradation?
Yeah, well, in this particular study, the TMA, we had a fecal TMA response, and so I'm sort of perusing the data here, and I'm looking for the TMAO, and you definitely see the increase in response to the Abomasal infusion in urine, and so that is sort of a good, it's about roughly 5% difference, maybe 10% difference relative to the other treatments, but it's certainly significant. And in that particular case, yeah, that's abomasal, right? That's post-ruminal. That's all coming from the role of TMA lyase past the rumen. So, yeah, there is a role there. You will see it in urine, and we showed that before, that 99.9% goes in the urine, and we didn't see that response in the milk. We did not see a change in plasma TMAO response in this particular study, but we have in others.
So how early in lactation were the early lactation cows?
They were ahead of a mean sort of time of 97 days in milk.
So can you speculate on what you may have seen if these were actually transition cows?
I would have suspected that, I want to be clear that most of the studies that we've done in the past that I can think of, I'm looking at the age of the late lactation cows, 270 days in milk. But an abomasal infusion, typically, I would have said, increases the choline levels, but earlier in lactation, the greater the response if it is bioavailable. But I want to emphasize that typically when you do these studies on the equivalency of choline ion across treatments, an abomasal infusion of choline, we've done this in mid-lactation before, and you get a choline response. It's very typical. I would have expected an increase in at least milk phosphocholine concentrations earlier in lactation, perhaps greater than what was observed in this particular study. I do want to emphasize here that the plasma choline response was enhanced, so not the milk in late lactation animals, but in the plasma choline response, we had more plasma choline in the abomasal infusion treatment relative to the others, but that was the same whether it was early or late lactation. It was only specific for the milk, where the early lactation animals had greater milk choline levels.
Joe, you started out by saying that, measuring choline bioavailability is very difficult. Have you found that method yet, or is there still more work to be done?
Oh, we found it. It is the other abstract we're talking about with Tanya France. It has its own limitations, which we'll talk about. That said, I think we're getting closer. I think one of the things I learned in this exercise is that, we have the ability to look at choline levels in fecal. When you look at rumen-protected choline products, that's a difficult thing to do. One, they produce a lot of feces, and so then you're trying to develop a proper sampling protocol to ensure that what you're measuring in that feces is the actual measurement, right? Because you're taking something, you're feeding something at these micro inclusion levels, and then you're trying to, see if it's actually ending up in the feces. That is a real challenge. I worry that potential limitation of our study is that we could be, we're providing an estimate. I'm not sure if, I think we have the potential to be underestimating just depending on our sample protocol.
All right. Well, we'll be looking forward to the next segment with Tanya. Charlie, I want to thank you for joining us today. I understand that you're a PhD student at Cornell. What's going to be your next steps here? Do you have some more research to do before graduating?
Yes, I have a big cell work coming up. It's basically investigate how ceramide induce insulin resistance in myotubes, from sheep, ovine myotubes.
All right, very well. Well, good luck with that, and thank you again for joining us. Joe, thank you as well.
Thank you.
Thank you all.
Right, in this segment, we're going to review an oral presentation given at the ADSA this year, and the title is The Metabolic Fate of Deuterium-Labeled Choline in Gestating and Lactating Holstein Dairy Cows. That presentation was given by Tanya France, and Tanya, welcome back. This might be your third time back at the Real Science Exchange, I believe, so welcome back. We appreciate you coming to visit once again. And I will say that we must say that Joe in the previous segment kind of gave us a prelude to this segment. So I'm looking forward to it because he said that you guys have come up with a viable way to measure choline bioavailability. So he kind of teased out a bit for us. So looking forward to this. Tanya, why don't you just kind of give us an overview of the study and the objective?
Yeah, so we were really interested in looking at the metabolic flux of choline in the dairy cow because, as we know, choline can be metabolized via two different pathways. And so in order to do this, we used stable isotope methodology, which has been well established in human and mice models. And we did this using this deuterium-labeled or D9 choline. And so we were able to measure that, the D9 choline, and the different metabolites. So if it was D3 or D6 choline, it would be utilizing the methionine cycle or one carbon metabolism, whereas if it has a D9 attached to any of the metabolites, it would be using the CDP choline pathway. Such that it would not be donating any methyl groups. So our hypothesis was that physiological state, so either late gestation versus early lactation, will influence choline metabolism, including the methyl group utilization in the dairy cow.
Tanya, since we can't see the pathways and so forth, what do you mean when you say D9 labeled? So
that means that there's 9 deuterium atoms which are replacing the hydrogens on each of the 3 methyl groups of the choline molecule. And so if a methyl group is being donated, it would lose one of those 3 deuterium atoms, and that's how we can measure it.
Perfect. You know what makes this study, so unique is that, again, you have these methyl groups that are on choline, and there's 3 of them. And so when you use this kind of tracer technique, you can sort of understand where each methyl group is going if it's labeled. And that's a powerful tool to understand how choline is converted into those other metabolites. And then when you can quantify them in different pools, then you can provide some strong estimates for how dietary choline is utilized.
Were there any surprises once you saw where the choline's being utilized?
Tanya?
No, I mean, I guess I was glad that both pathways were being used during both late gestation and early lactation, but I wouldn't say I was surprised by that.
Well, I was sort of surprised. I mean, I was certainly pleased to see the responses that we saw. Again, there were so many big questions surrounding choline biology that I think this study has answered. And in reality, the study itself is a rather simple study and relies a little bit on the use of mass spectrometry, obviously. But, to provide definitive evidence that choline is a methyl donor is something that we talk about it. We say, hey, it's a methyl donor, but you really need some concrete evidence once in a while. And this study does that. It actually shows that it is indeed behaving as a methyl donor and providing methyl groups within the methionine cycle. So that's something we wanted to see, and we saw it. And then, it's nice when Tanya mentions that being able to look at the pathway utilization, and we certainly gained a lot of value there to try to understand that. I think there's so many other things. I mean, I don't want to steal the thunder away from Tanya. Tanya's the one who specifically discovered this, but one of the cool findings is that we discovered. So we wanted to better understand the post-ruminal degradation of choline to trimethylamine and TMAO, and one of the metabolites that we discovered was D3 trimethylamine. And not getting too sciencey for everybody, but basically this provides evidence for this choline recycling that can occur. That choline enters the body of the animal. It can be act as a methyl donor. It can be converted back into choline-containing phospholipids, and then those phospholipids could be entered bile and go back into the intestines and be acted upon again by the bacteria post-ruminally. And it's just we provide evidence for this recycling, direct evidence for it, and it's certainly something that we read in textbooks about before, but it's nice to actually be able to measure it and see it.
Are there any follow-up studies then, or any plans now that you've got this tool? How do you plan on using it?
Well, I think you're bypassing some big stuff here. Okay.
All right, sorry.
This study has some major conclusions that we can't necessarily ignore, and we really wanted to try to look at how much of the choline is being used in these different pools. And I don't have the numbers exactly in front of me, and Tanya, you can correct me if I'm wrong, but depending on stage of lactation, we're seeing anywhere from 5 to 9% of the choline that gets post-ruminally is getting converted to trimethylamine and TMAO. And so something as high as 9 or 10%, that is certainly, I think, significant when we're trying to provide some estimates or thinking about metabolizable choline supply. And we need to remember that, and we need to think about that moving forward and try to develop approaches that are less costly than this study to allow us to include that as a potential correction when we want to calculate bioavailability. The other one was on how much of the choline was actually used by the mammary gland. And so it depended on the metabolite involved. And I think we estimate around, I think it was 12% of the choline that was infused was actually used by the mammary gland for milk choline. But when you added up all the metabolites, so I think, Tanya, what was it, 24?
It was 25%.
Was being used by the mammary gland. So that's significant, and it just emphasizes that it's not just choline, that it's all these other metabolites that we need to think about.
Were there differences in the late gestation cows versus early lactation?
So we did. I mean, Tanya can provide some comment here, but we did see changes in the plasma concentrations of these metabolites, which is what we would expect. We did see in numerically lower milk choline, plasma choline concentrations, but one thing that I think was really important here is, again, we saw evidence for that methyl group utilization by the methionine cycle, and we see that the metabolites that were involved in that PEMT pathway, those metabolites are increasing sort of early lactation. Some of these p-values are significant, others are tendencies, but overall, it looks as though that there is an increase in the utilization of choline as a methyl donor in early lactation. That said, it didn't say that the other pathway wasn't important. It was also important as well. And I think when you look at the non-ruminant data, there's plenty of evidence to suggest it actually plays a much stronger role in providing phospholipids. But it's a combination of the both that appear to be very relevant in an early lactation animal, or important for that animal.
All right. Anything else, Clay?
So now I think it's time to ask what's next.
Now I can ask that question.
What do you want to do next, Tanya?
I mean, I think a big part of it that we were unable to measure and account for was how much of that choline we infused, how much of it was going to the fetus. So there's a lot of evidence of that happening in humans, and they were able to do that with D9 choline, and there was a large part of the PC that was made that had the D9 D3 attached to it going towards the fetus. And so that is something that we weren't able to account for in this study. However, I think it would be really cool if we were able to somehow do that in the cow. It would require infusing this leading up to and while they calve in. But
so how many days were the infusions?
They were 5 days.
And
Yep.
How long did it take for the concentrations to peak during that 5-day period?
At day 3, we saw it kind of start to plateau. So we measured the last 2 days, so day 4 and 5, but over the course of time, it started at about the 3rd day.
In terms of, I mean, I have so many ideas, but I'm gonna need a massive budget to start to answer some of these. If anybody wants to look at the cost of a labeled choline, you'll understand why it hasn't been done before. But that said, yeah, it's such a powerful tool, and I think there is opportunity to, answer some other questions about choline utilization and their interactions with other nutrients and other forms of life stages of the animal, environmental stresses, things like that. There's a lot of opportunity there to answer some big questions, relatively quickly if we have some investment to support it.
Okay. All right. Very well. Want to thank everybody for joining us today. This has been quite interesting, and looking forward to the next time, Tanya, if you'll be joining us once again.
AminoSure XM Precision Release Methionine, the next generation in amino acid balancing. With AminoSure XM, you can save up to 5 cents per cow per day on your methionine investment. Try it today and receive an additional 2.5 cents per cow per day savings with Balchem's limited time rebate offer. Contact your Balchem representative to learn more.
And we're back, and in this segment, Clay and I will be reviewing an oral presentation that was given at the ADSA. The title of that talk was called The Effects of Dry Period on Heat Stress and Rumen-protected Choline on Productivity in Holstein Cows. And that was presented by Maria Torres de Bari. Her advisor is Dr. Jeff Dahl from University of Florida. And Jeff, this is not your first time at the Real Science Exchange, so welcome back. Appreciate having you back here.
Thanks, Scott.
Would you mind kind of introducing your student and telling us a little bit about her?
Sure. Maria is a master's student with me. She did an internship a year plus ago with me, which is usually sort of the first battle testing of students. And she survived that and decided that she would agree to come start her master's. She is a native of Brazil, is a veterinarian from Brazil, and has been working on this project just about a year now.
Okay, excellent. Well, Maria, welcome. It's nice to have you here at the Real Science Exchange. Why don't you start off by telling us the objective of the study, and then what were some of the methodologies that you used in the study?
So as our objective in our study, we know, as we saw in other research, the heat stress has a negative effect on the memory growth in dry cows comparing to the cooling cows. And when you see some other research about the cooling effect on, for example, memory growth in vitro studies, we see the choline could reverse the growth of those memory epithelial cells. So, as our hypothesis, we'd like to test if the supplement with choline during the entire dry periods in multiparous cows could reverse the negative effects of the memory growth and function in the cows.
All right, very well. And what was some of the methodologies used? How did you do this?
So we had 51 multiparous cows that we enrolled then around 47 days before calf. So in the day that they dry it off, we enrolled in the trial, and we split them in four treatments. Cooling and heat stress, both receive RPC, that is Rumen-protected choline, or control. So for the cooling group, we provide to them shade, soakers, and fans, and for the heat stress group, we just provide shades. During the entire dry period, you have dry matter intake collection daily, and also respiration rate and rectal temperature. Once they calve, we stop with the supplementation with choline, and we keep them in the trial until 60 days in milk, and we continue tracking the dry matter intake and the colostrum yield and milk yield during those 60 days.
So Maria, at calving, were the cows still cold or heat stressed? How were they? How were those treatments applied then?
We haven't had a treatment, a temperature treatment. We keep them in a cooler pen with fans, soakers, and shades.
Okay.
So during lactation, the cows were all cooled.
Okay. And did I understand correctly that they did not have choline after calving? It was all pre-fresh?
Yes.
Correct.
Yeah, okay.
But for the entire dry period.
Okay. Yeah.
And what is the period of heat stress in Florida?
Forever.
That's right.
I was curious as well. Were you documenting the heat stress index with this as part of the study?
Yes, we have the data from the THI, but they were not different among the control and among the heat stress and the cooling, and the cooling side was 72 and the heat stress side was 73, the THI.
Essentially, between June and October, we don't have an hour that we would have a THI less than 72.
We've only got partial results so far from the trial. Is that correct? Has the trial ended?
Yes, we finished the trial, but we haven't taken a look in all the data that we have yet.
Okay. And so you just presented some partial data at the ADSA this year then?
Yes, we have like next analysis that we want to perform as a cortisol analysis. Also, we performed mammary epithelial biopsies. And we also want to take a look on that to see how is the rate of proliferation and apoptosis in those cells for our cows comparing the treatments. And also we want to track the calf development data. So we also have biological material from calves that we want to take, we want to analyze.
So Maria, what are the results so far that were presented at the ADSA meeting?
As we were expecting, we saw a negative effect on respiration rate and rectal temperature in the heat stress group. So they have a higher rates for those variables. The heat stress compared to the cooling, they eat less. I mean, they decrease their dry matter intake. Also, they had a shorter gestation length. The calves were born lighter compared to the cooling animals. And the dry period was shorter compared with the cooling animals as well. Heat stress with the cooling animals. And the milk yield, we have a decrease in milk yield in energy corrected milk. We have a negative effect of heat stress, decrease in the energy corrected milk compared to the cooling cows, compared to the cooling group.
Am I correct? About a 6 kilo difference in energy corrected milk yield?
Yes, about 7 kilos.
7 kilos.
Yes.
And that was for the heat stress versus cooling as main effects. Now, the choline did what?
The choline, when you compare the cooling cows that were treated with the choline, they produce 2 kilos of milk more than the cooling cows without supplementation.
And what about the heat stress cows?
So we had the heat stressed cows. The heat stressed cows supplemented with choline. You had a decrease in milk production compared to the heat stress control group. So what do we see for the energy corrected milk when you compare the heat stress with the heat stress that were supplemented with choline is the choline didn't combat, the choline didn't mitigate the negative effects of the heat stress in terms of milk yield. And when you take a look on the control group for cooling and the animals that were supplemented with choline, just a minute. Mm-hmm. So when you take a look on the heat stress cows that they were supplemented with choline, we didn't see a reverse compared to the control heat stress control group. We didn't see a reverse in the negative effects of the heat stress in terms of milk production. But when you take a look on the cooling cows compared to the cooling cows that receive the choline, the cows supplemented with choline had an increase in 2 kilos of milk production milk daily of in terms of energy corrected milk.
And that's a really good finding because this was only prepartum feeding of the choline.
Yes.
In this case.
Prepartum feeding, yes.
And we are increasing our numbers because that is trending the right way. It's trending towards a positive effect of choline, but we need to increase our numbers to get a better estimate.
Mm-hmm. And I think, Jeff, part of the main objective of the study is to take a look at the mammary development, and you've done the mammary biopsies, and so that's kind of exciting to see what that data is going to look like. When can we expect to have that data available to the public? Was that something we'll present next year at the ADSA?
Yes, we are. We want to start to work on this as soon as possible. I just started a new trial to have more data. The same as I mentioned here, we are doing a new trial with less amount of animals. But as soon as I finish this one, the plan is to start with the memory epithelial cells analysis, and then to present in the ADSA next year.
Perfect. Any other questions from you, Clay?
So what's any take-home messages at this point based off of what you're seeing so far?
Well, I would say the practical implication is if you're not cooling the cows when they're getting choline, you may be not maximizing or optimizing your benefit. So it's not a silver bullet to overcome effects of heat stress by any means, but if you don't cool them, you may not be getting as much of the advantage or any advantage to that. The other important part, I think, is the calf. Right? What are we seeing with the calf? And it appears, at least from our initial look at the data, that there is a reversal of the negative impacts on the calf with the choline supplementation to heat-stressed animals. So if that holds up, then that in itself could be a huge practical implication.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email at anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars takes place on the first Tuesday of every month, with the top research and nutrition topics that will impact your business. We also include small ruminant, monogastric, and companion animal-focused topics throughout the year. Visit balchem.com/realscience to see the upcoming topics and to register for future webinars. You can also access past webinars and search for the topics most important to you.